Hello students, welcome to Distinction Learning Platform, and on this platform you find revisions, test quizzes, a lot of stuff. So before we begin our lessons, make sure that you subscribe so that you can be updated with upcoming videos. To make the story short, let us be. Don't forget to like the video. In this video, we're talking about what? We're talking about nerve physiology, but this is the second part when it comes to nerve physiology. Remember that we talked about the first part, talking about what? Introduction, the structure of the neurons, the glial cells, classification of neurons, all those things. Make sure that you watch that video so that now you can be comfortable when you start talking about what is this part of the video. Okay, so in this part we talk about the axonal transport, we talk about the neuronal transmission, action potential, and most importantly what we talk about what the laws, which is maybe all non-laws, then the law of conduction, Bell-Magendi principle, the law of what summations, all those things we'll talk about them. So make sure that what you pay attention. Okay, so now let's start with the first part. Let's start with the axonal transport. So axonal transport, what does it mean? It means that okay, if you look at the structure of the neuron, if you remember a structure of the neuron like this, okay, it has this part which we call the soma, or sometimes it is called what? The cell body. Okay, this cell body, it has this long part which we call what? Axon. Okay, and it has this part, let's just call it axonal what? Axon terminal. Let's just say AT. Okay, soma, which is also called the cell body, axon, and axonal terminal. This axonal transport, it means what? It means it's a type of transport where there will be substances that will be made in the cell body, because the cell body is the one that synthesizes a lot of what the structures, proteins, neurotransmitters, everything. They're going to be made by the what? The cell body. Okay, so now these things, if maybe they are needed in the what? Axonal terminal, they have to be transported from this cell body, then going in this direction. Okay, this is the transport. Again, sometimes maybe if there's something that is needed by the cell body but it is here in the axonal terminal, what do you think will happen? This transport also what? Okay, going like this. Okay, so if you talk about the axonal terminal, axonal transport, what do we mean? We mean that it's a transportation of important substances, okay, coming from the what? Either from the cell body going to the axonal terminal, or maybe coming from the axonal terminal going back to the what? Cell body. Two important transport systems that you need to understand. Okay, so but before we go to this there's some important structures that you need to understand that are involved in the what? Axonal what? Transport. What are those? So let us look at this simple structure of the what? Neuron. This one, you've seen this. These are very, very important. What do you call those? We call them what? Rough endoplasmic reticulum. Also called, if you remember from the other video, we said that is also called missile body. So this is what we call rough endoplasmic reticulum. Let's just call it RER. Okay, rough. So those are what we call rough endoplasmic reticulum. What could be the function of the rough endoplasmic reticulum? It is the protein synthesis. It's the one important structure that makes what? Proteins. Okay, we need to know that pattern. Okay, what about this one? This red one, we know this one is the what? Nucleus. So nucleus, that's where you can find DNA. It's the one that's going to give instruction to this transport, a lot of things. Okay, that's all about that. Now, this is the whole thing here. This is what we call the axon. Okay, this one is what we call the axon, and this is what we call the axonal terminal. Sometimes they can call it synaptic knob, a lot of words that they use. Okay, but what I'm most interested is this red structure. What we call that red structure is what we call the microtubule. Very, very important structure. Microtubule. Okay. Microtubule is going to do a lot of things that is very, very important in what? The transport. Okay, on this microtubule, okay, on this microtubule there's what? These funny soldiers there that are there. So those fine structures. So these, they are what we call, so if you look at from this one, the black ones, we're going to call them what? What are we going to call them? We're going to call them what? Kinesin. So these black structures, these, okay, we're going to call them what? Kinesin. Okay, and what about these blue ones? We are going to call them what? Denine. Okay, we are going to call them what? Denine. Okay, we call them what? Denine. Okay, so now these two important structures, the kinin and the denine, they are embedded to the what? Microtuba. They are important structures that are going to us. They use what we call ATP for them to start transporting what? Substances. So what's the important function of kinesin and denine? This one, denine and kinesin, they are used in what? Transport. Okay, they are going to get some substances from the cell body or maybe from the axonal terminal, and they are going to start what? Transporting those substances. Okay, very, very important. Now, this axonal transport, there are of two types of axonal transport. Since we know the structure, now let's talk about the types of what? Axonal transport. What are those types? So there are two types of transport, as you can see. If the direction, if this is the cell body, okay, and this is the axonal terminal, if the direction is going like that, what do we call that type of transport? We call it what? Anterograde. Okay, what do we call this type of transport? We call it what? Anterograde. But sometimes they're going to use another name, which we call orthograde. So if they use orthograde or anterograde, it's just the same thing. Okay, they're going to use also this one, orthograde. Orthograde or anterograde, these are the just same words. Okay, they're just used to describe the type of transport that is coming from the cell body going to the what? Axonal terminal. What about this direction? Okay, what about this direction? What do you think? This direction is what? Retrograde. Okay, retrograde. Okay, this is the retrograde type of what? Transport. This one is going to carry what? Information, or it's going to carry substances from the axonal terminal back to the what? Cell body. Then that type of transport is what you call retrograde. Okay, now there's an important thing that you need to understand now. You see, I talked about the kinesin and denine. Okay, kinesin and denine. So this kinesin and the denine, they are involved in different types of what? Axonal transport. But if you look at the kinesin is used in what? Anterograde type of what? Transport. So here they are going to use what? Kinesin as one of the important what? Substance to transport. One of the important enzymes to transport what? These substances that are coming like that. Okay, and retrograde is they are going to use what? This one, this type of enzyme, which is denine. Okay. Okay. Now we proceed. So now, how does that happen? Remember that now let's talk about the important substances that are going to be transported in anterograde and the important substances that are going to be transported in the what? Retrograde. So let's start with what? Retrograde. This one. Okay. Let's start with this one. So let's try to imagine to say this is the direction that we're talking about. The retrograde, it is going like that. So this is the retrograde. Okay. Oh, I mean this is the anterograde. This one, the one that is going from the cell body going to the axonal terminal. Let's start with that one. Okay. But there are two different types of anterograde. So let's say this is the anterograde. Okay, this is the anterograde. Okay, there are how many types? There are two types. Okay, there are two types of anterograde. There's what we call the one which we call the fast. Okay, fast conducting anterograde. So the one that is fast, so we call it fast anterograde type of what? Axonal transport. So it is fast. Why? If you look at the velocity or the velocity on how it conducts those what? Those substances from the cell body going to the axonal terminal, it has been talked about. It is about this is the rate about 200 millimeter per what? Per day. To what? 400 millimeter what? Per day. Okay, per day, like that. Okay, so this is the first one. So it's between 200 to 400 millimeter per what? Per day. So that's how fast it can transport those substances in a day. Okay, that's what we call fast. But the slow one, so sad about this one. The slow one, basically the slow type of anterograde transport is about what? It's about 5. This is very, very slow. 5 to what? 10 millimeter per what? Per day. It's very, very slow, this one. Okay, and it's so sad. So what are we saying? Under anterograde type of axonal transport, there is what we call fast type of conduction, or fast type of what? Anterograde type of what? Transport. and there's the one which is slow. If you look at the numbers here, this one is between 200 to 400, and they just say 400 millimeter per day. This one, the slow one, is what? 5 to 10 millimeter per what? Per day. That's how slow it can what? Do or conduct what? Or transport the substances. Now the question is, what are those substances transported in anterograde type of what? Transport. What are those? Okay. What are those? So substances, so let's just say molecules or substances, What are those? Here, let's start with what? The first one. The first one is what? The first one is what? Proteins. Okay, remember that this half endoplasmic reticulum is going to what? Synthesize proteins. If it makes proteins, those proteins, if they are needed in the axonal terminal, what do you think? They'll start coming from the cell body and they'll start going to the what? The axonal terminal using what? Anterograde type of what? Transport. Okay, that is the first one. Proteins. What else apart from protein? What do you think? Neurotransmitter. Okay, neurotransmitter. So we talk about the neurotransmitters. So neurotransmitters, these are the important substances. So neurotransmitters, these are the important substances that helps to what? Transport what? Nerve impulse from one neuron to another, or maybe from a neuron to what? To a muscle, or to an effector, or maybe to a gland. Okay, so those are important neurotransmitters. We'll talk about the types of neurotransmitters. Don't worry about that. Okay, we'll talk about that. So also neurotransmitters, if they are made by the what? Rough endoplasmic reticulum, they're going to be transported via the what? Anterograde type of what? Transport. Okay. So we've talked about this anterograde. Let's talk about now retrograde. Okay, retrograde, this one. Let's talk about it. So the one that is going to what? From the axonal terminal going to the what? The rough endoplasmic reticulum. Okay, before we go from this side, so let us look at how it does that. Okay. So if this is the rough endoplasmic reticulum, it's going to synthesize. Let's just call this one as what? Let's just call it proteins. Okay, these proteins. Okay, these are proteins. Then what happens is that for them to be transported, these proteins, okay, these proteins are going to be put in the what? Vesicle like that. They're going to put in the what? Vesicle like that. Okay. So my God. Okay, they're going to be put in the vesicle like that, and after putting them into the vesicle, they are going to be added on this important what? Structure. What do we call that? We call it what? Kinesin. Okay, so they are going to be put on in this kinesin so that now it can be transported like that up to where it's going to reach where? The axonal terminal. This is the protein which is here. Okay, the moment it is here, what do you think is going to happen? Either maybe it's going to be secreted, okay, from this cell so that it can be here, or maybe to be just left in the axonal terminal. That's how that happens. But for this to happen, for this kinesin to start doing their work, what do you think? They're going to start using what? Energy. So no wonder you need to understand to say axonal transport, it will depend on what? ATP. Make sure that you understand it's an active process. Okay, so let's go to this one. So this one now, this one was saying it's going to depend on what? This type of what? Enzyme, which is dynein or denine. Okay, denine, this type of enzymes, these are very important. So remember that now they are going to make sure that catalyze this type of direction. They are going to get information or get substances into this, and they are going to transport them via what? Denine molecules attached to the what? Microtuba. Okay. Now, what do these substances that are coming that are what? Transported in what? Retrograde type of what? Transport. What do they come from? They come from where? Outside. Okay. They can come from maybe let me just say which we call, so there are these substances. So examples of these substances, so let us use blue. Examples, or let's just say molecules. So molecules that are transported using what? Retrograde type of transport. What do we call that? Let's start with a very dangerous one, which is a virus. What is that good example of that virus that is transported via the retrograde? Rubella virus. That is very, very important. Okay, Rubella virus is going to be transported via the what? Retrograde type of transport, and it will start causing what? Infections, which is very bad. Okay, what is the other one? It's not always something that is bad. Also maybe some growth factors. Okay, some growth factors. So we'll talk about what it means in other lessons, what it means when we'll talk about the growth factors, okay? Also growth factors are going to be transported retrograde, so that they go to the they go to the cell board and they promote maybe the development of those development of the neurons, also modulating other important functions of the neurons. So growth factors are needed by the neurons as well, okay? Those are the important things. Now, what else are we supposed to know? So this is all about the axonal transport. Okay, this is all about the axonal transport. Remember that it's also this molecule, whether growth factor or virus is going to be put in the what? It's going to be put in the vesicle as well. This is a vesicle up until it reaches the cell board. The moment it reaches the cell board, now it's going to be put into something which is very important. Okay, put into something which is very, very important. So this substance, which is either growth factor virus, the moment it reaches the cell board there, it's going to be put into another important vesicle, which we call lysosome. Okay, this lysosome. So lysosome is very, very important. It's either since it was a virus that was coming here, let's say, imagine to say it's a virus, because the one that is going to be transported using this, then if it reaches the cell body, inside the lysosome there, that's why you are going to find a lot of substance that can kill a virus, either maybe bacteria, anything. So like enzymes, processes, they are there. Elastases, they are there. So they are going to start killing that bacteria. Or maybe if you had a mitochondria, remember that under here you had some organelles that were damaged here. They're going to be transported back to the cell body, and maybe they're going to be destroyed in the lysosomes. Okay. So no wonder that they sometimes say this type of vesicles are what we call recycling type of vesicles. You are just recycling, okay? Recycling substances that are used there. If you are recycling, let's say you get a mitochondria, then you take it back to the cell board, what do you think will happen? It will be recycled. It's going to be reused, okay? Like that. But in case of the virus or bacteria, they're going to be killed by the lysosome type of vesicle. Okay, boom, boom. Let's go to another one. Okay, let's go to another one. So let's go to this part again. So we're almost there. So let's go to this part. So neuronal transmission. Now you need to understand how electric impulse move from one nerve cell to another nerve cell. You need to understand about that. Okay, very, very important part. If you talk about the neuron transmission, we need to understand about very, very important things. We'll talk about now the neurotransmitter that I was supposed to talk about. Okay, we'll talk about the types of neurotransmitters. Okay, so let us start with this one. So as you can see, there are two neurons here. Okay, let us try to name something that it's going to be necessary for us to understand fully. Okay. So we have this neuron part. Okay, this neuron part, as you can see, I'm just taking this last part, which we call the axonal terminal, like that. Okay, so that we understand what happens there. Okay, this is the axonal terminal, and this is another axonal terminal. Okay, this neuron that is starting, what we call the type of a neuron that is starting. So let us understand about that. Okay, I'm going to remove this. So we need to understand something here. Okay, we need to understand about something. So what we call this type of a neuron, so let's say we have two types of neuron. We've got this neuron, okay? We have another neuron here, okay? These are the two types of neurons that we're talking about. The neuron that is first stimulated and conducting electric impulse like that, okay? What we call that? This is the first neuron. The first neuron is called what? Presynaptic neuron, okay? This is a what? Presynaptic. So I'll tell you why I'm using synaptic. So this is a presynaptic neuron, and this one after the synapse is called what? Postsynaptic neuron. Okay, and remember that the gap between these two neurons, what we call this? We call it what? Synaptic gap or synaptic cleft. So we are saying this one is a synaptic what? Cleft or gap. So the space between two neurons is what we call synaptic cleft or synaptic what? Gap. And if you remember that the first neuron that's going to be stimulated is called what? Is presynaptic. The neuron that's going to be stimulated after the synaptic gap is called what? Postsynaptic what? Neuron. You need to understand about that. Okay? Now, since we've understood about those terminology, and this space we know now, this is a what? Synaptic what? Cleft or synaptic gap, and this is a presynaptic neuron. So let's just call it. This is a what? Pre, and this is a what? Post, so that we fully understand postsynaptic neuron and presynaptic neuron. Okay. What other important things? Let's understand something. Again, the other important thing is what? Here we have a what? A vesicle there. Okay. We have a vesicle. This is a what we call what? A vesicle. Okay. That is a vesicle. Remember that if you come back here to put everything together, this vesicle came from where? Where did it come from? It came from where? It came from the cell body there via two different what? Axonal transport. Remember that the retrograde and what also the anterograde type of what? Axonal transport. But basically this one, for it to reach the axonal terminal, it came via what? Also grade or anterograde type of what? Axonal transport. Okay. Now inside this vesicle, there is an important substance that you need to understand. Inside this vesicle, there is what? Important substances that we call what? Neurotransmitter. Let's just call them what? NT. Neurotransmitter. NT. Okay, inside those vesicles. Okay. Then what else? What are we supposed to understand? What do we call this membrane now? So this is what we call the axonal terminal membrane. Okay, we'll talk about that. Anyway, now another thing. So it's a lot. Another thing. So if we talk about the neurotransmitters, okay, so these neurotransmitters, the one that are inside here, we got different types of what? Neurotransmitters. So before I start talking about anything, let's start understanding what we mean if we say a neurotransmitter. What is a neurotransmitter? A neurotransmitter basically it is what? A substance that is used either to stimulate a neurotransmitter is what? It is a substance that is used to what? That is used to transport what? Nerve impulse from one location to another. Sometimes neurotransmitters can be what? Can be used to inhibit the neurotransmission. So we've got important types of what? Neurotransmitters. So the first one is what? Let's just call them excitatory. Let's just put them in full. Excitatory neurotransmitter. Okay, excitatory neurotransmitter. Basically, these they are going to stimulate, so they're going to promote what? Neurotransmission. There are also others which we call inhibitory. Okay, inhibitory neurotransmitter. These they are going to what? To inhibit the neurotransmission. This transmission that we talk about. If you have a neurotransmitter that is what? Inhibitory. Basically, it's going to what? Prevent the neurotransmitting. And there's also another neurotransmitter which we call modulator type of what? Neurotransmitters. So there are also what we call modulatory transmitters. There are also those. Now, excitatory neurotransmitters, what are the good examples of those neurotransmitters that likes to what? Promote the nerve impulses from one location to another, from one neuron to another? What are those? We'll just talk about a few. Okay, we have what we call glutamate. Okay, what else? Acetylcholine. Okay, so two important what? Excitatory type of what? Neurotransmitter. There's what we call glutamate. So this is glutamate as well as what? Acetylcholine. Inhibitor neurotransmitter, these they are going to inhibit what? The neuronal transmission. What do we call those? There's what we call GABA. So we just call it what? GABA. Okay, so in full we call it what? Gamma aminobutyric acid. So it's better we just call it what? GABA. Okay, GABA is an inhibitory as well as what? Glycine. Okay, but the most important one is what? GABA. Modulator type of neurotransmitter, basically we can just get another one, acetylcholine. Okay, acetylcholine can act as what? Excitatory and as well as what? Modulator. Very, very important. Okay. Now, since we know the types of neurotransmitter, now let's talk about what the neurotransmission, okay? So let's say here, either way, this vesicle has what? A certain neurotransmitter, whether excitatory, inhibitory, or maybe a modulator like that, either way, okay? What do you think is going to happen? Here, what will happen is like this. The action potential will come like that to this what? Synaptic what? Presynaptic type of what? Neuron. The action potential will come down like that in that direction. It is coming in this direction. Okay, it is coming from the cell body there to the axon up to the what? Axonal terminal. It's coming like that. Okay. Now, if that happens, what do you think? The moment it reaches the axonal terminal, something interesting is going to happen. Which is what? It's going to open up something which is very, very important. What is that something? They are what we call gated channels. Okay, they are what we call what? Calcium gated what? Channels. So calcium gated what? Channels. Calcium gated channels. Okay, this is a calcium gated what? Channels. So the moment these action potential, we'll talk about the action potential. The moment this nerve impulse or action potential reaches the axonal terminal, it's going to open up the what? The calcium gated what? Channels. So if it opens up these calcium gated channels, what do you think you start rushing into the axonal terminal? This one. Okay, it's this one. So the calcium from outside here, it will start what? Going into the what? The cell here. Okay, that's what will happen. So the calcium ions will start now rushing into the cell. Why? Because the nerve impulse reached the what? Axonal terminal. So this will happen like that. The moment now calcium accumulates in the axonal terminal, then what do you think will happen to this vesicle containing the neurotransmitter? Then this vesicle containing the neurotransmitter, they are going to start, they will fuse with what? It will fuse with the axonal terminal membrane. It's going to fuse with this one. Okay? So it's going to fuse with that one. So fusing basically is going to release, as you can see inside here, a vesicle is what? A neurotransmitter. So this neurotransmitter is going to be what? Release on the what? The synaptic gap there. So that's what happens. Then next, the other important thing that will happen is this one. Remember that this is a postsynaptic neuron. It has what? Receptors there. Okay, it has what? Receptors. Receptors, it means that these are the things, or these are the important structures that are going to receive what? A neurotransmitter. So this neurotransmitter that has been released on the synaptic cleft is going to bind on this what? Oh my god. It's going to bind on this what? Receptors. They're going to bind on this what? Receptors. Okay? The moment now these neurotransmitters bind on this receptor, this one this postsynaptic neuron is going to be what? Activated. Thereby what? Transmitting the what? The action potential, the nerve impulse. So that's how what? Neuronal transmission happens from one neuron to another. These two neurons, they are not connected to one another. There is a gap there. But how do these nerve impulse or electric impulse from this neuron jump to the which one? The postsynaptic neuron. How? It's because of this mechanism that we've talked about. Okay. The moment this neurotransmitter binds to these receptors, they are going to start activating now this postsynaptic neuron. Thereby the action potential is going to happen. That's how it happens. Okay. So, but we'll talk about the other important thing that you need to understand. The question that you can ask is that, okay, after this neuron has been activated, this neurotransmitter that has binded to the what? To the receptors, where does it go? Now it will depend on the type of what? A neuron that is a type of neurotransmitter that is binding to this receptor. So, but basically the most important one is what? Acetylcholine. If this is acetylcholine, which is one of the what? The neurotransmitter, then what's going to happen? It's going to be broken down by certain enzyme on the what? Synaptic cleft. We call it what? Acetylcholine what? Esterase. So acetylcholine esterase is the one that's going to start breaking down the what? This is what? Acetylcholine neurotransmitter. So that now this postsynaptic neuron cannot be further stimulated. Okay, you need to understand about that. Okay, so this is all about the what? Neuronal transmission. Now let's go to another point. So let's go to this one, action potential. Okay, so here, since we've understood something about this, okay, make sure that you've taken note of this. Now let us understand about the action potential. Okay, let us understand about the action potential. So it is a very important topic when we talk about the nerves physiology. So you need to be aware of the action potential, resting membrane potential. You need to be aware of that. Okay, so let's talk about the potential of these cells. So let me just remove everything here. Okay, removing everything so that you can fully understand about this. Okay. So action potential, let me draw something. Let us start speak in cell. Okay, let us start speak in cell. Okay. So we're understanding about the action potential and the resting membrane potential. Okay. We know that potential, so resting membrane potential or action potential, we're just talking about the potential difference across the cell membrane. That's all about what the resting membrane potential and what the action potential. So the potential difference across the cell membrane, okay, and this is usually measured in what, millivolts. So it's usually measured in what, millivolts. Very important, because those are the smaller type of potential difference that is there. Okay, no wonder they are measured in what, millivolts. Okay, and they are very fast. Okay, now so if this is a cell, basically let's start talking about the first important one, the resting membrane. So let us understand about this one, potential. Okay, if we understand about the resting membrane potential, we can understand about what the action potential as well. So resting membrane potential, what is it? What is it about? So resting membrane potential is a type of potential difference across what, the cell membrane, okay, when a nerve cell is not stimulated. So if you are talking about the resting membrane potential, it's a type of potential difference that occurs in what, in cells that are not what, is stimulated. So basically we're talking about a nerve cell. If a nerve cell is not stimulated, it's going to have a certain potential difference measured in millivolts, and it is what, if it can only happen when what, the nerve cell is not stimulated. Okay, you are not stimulating the nerve cell, so that's when this can happen. Okay, now what are the important factors contributing to that? So let's start with the first number. So what is the normal number of the resting membrane potential? It has been shown that for the nerve cells, it is about what, we know this one, negative 70 millivolts, like that. We'll talk about why it is negative. So you need to be understand something like that. Okay. So what are the important factors contributing to this resting membrane potential? The type of membrane potential where what, a cell or a nerve cell is not what, is stimulated. Okay. So these are the things. It has been shown that when a cell is not stimulated, the outside potential, the outside what, charge, the outside of this bilayer type of what, lipid layer or the cell membrane, this outside of the cell membrane, it is what, positive, and inside it is what, negative. Okay, and this resting membrane potential, it has been shown that it is what, the type of potential happens across what, across this cell membrane. Okay, now since we know that like that, so now the question that we can ask is that why inside the cell is what, negative? Why is it like that? Why is it negative? The important answer for that is this one. The reason why it is negative, if you look at the ions, so you need to understand about the what, the ions. Okay, ions that are very important. Ions like what, sodium, ions like what, potassium, ions like what, chlorine, and also proteins that are what, charged, let's just say charged what, proteins. Okay, so if we're talking about why it is negative inside the cell and why it's positive outside the cell, we need to be talking about the ions like that. Okay. Okay. Now, if we talk about the sodium, let's say outside. So here, the sodium which is outside and the sodium which is what, inside. So here, sodium which is inside. What do you think? Sodium is what? How many sodium is outside? So sodium is what? How many sodium ions are outside? So outside here, sodium outside is what? Is a lot. So many. Okay, so sodium outside is many, then here it is what, low. Okay, inside the cell, so the amount of sodium that is inside the cell is what, low. Then what about potassium? Outside the cell, potassium is what, Okay, what do you think potassium outside? Outside potassium is what? Low compared to what? Inside it is what? A lot. It is many. Chlorine or chloride is more like potassium, or it's more like sodium. Here chloride is what? If you talk about outside, it is what? Outside it is many. Then inside it is what? Low, like that. Okay. But if you talk about the charged particles or charged proteins, charged proteins, outside the cell they are what? Low. Then inside the cell is what? They are what? Many, like that. Okay. Now, if you understand something like that, sodium outside is a lot. Outside this cell it is a lot. Inside it is what? Low. Potassium, it is just the opposite. Chlorine, it is same as what? Sodium. Charged proteins, it is same as what? Potassium. Like outside it is low. Inside the cell it is what? It is a lot. Okay. Now, the reason why the inside is what? The inside of this of this cell is negative, it is because of this. So the first important reason is what? This one. Okay. It is this one. Okay. So what we call these channels? So these channels are what we call potassium channels. Okay. These are what we call, this is a case potassium channels. If you look at potassium and sodium, it has been shown that this cell membrane is more permeable to potassium compared to sodium. Why? Because this cell membrane has a lot of potassium channels. Okay, if it has a lot of potassium channel, what do you think? Potassium will start moving out like that passively. Why moving out like that? Because if you look at potassium inside the cell, it is a load. If it is a load, it can passively go out while it is what? Low. Okay? Make sure that you understand. And potassium is what? Positive. Potassium is what? Positive. If positive charge particles are moving out of the cell, what do you think is going to be left? Negative. Okay, that's the first reason why this one is what? Why this one inside the cell is what? Negative. What's the second reason? Another second reason, important reason. Okay, we've got this important what? We've got this important transport. Okay, that important channel. This channel is what we call sodium, potassium, ATPase. Okay, this one. Sodium potassium ATPase is also one important channel. One important channel that is responsible for what? The negative charge inside the cell. Why? If you look at here, what it does is like this. Okay, it's going to pump sodium, 3 sodiums out, and it's going to pump what? Potassium, which is about what? 2. So 2 potassium inside and 3 sodium out. As you can see, how many ions are moving out? They are about what? 3. How many ions that are going in? They are about what? 2. Remember that you are moving a lot of ions from the what? From the cell. Then if you are moving out a lot of ions from the cell, what do you think? The inside of the cell is going to be what? Negative. Okay. So that's the main reason why we're talking about that. Okay. So now, since we've understood about that, we've understood the reason why it is negative inside. So if we try now to measure everything, we're going to make sure that you are going to have negative 70 millivolts. Now, let's do another cell. So this is all about what? The rest membrane potential. Now let us understand about the what? Action potential. Okay, let's talk about the action potential. So action potential now is going to depend on different types of channels. Okay, different types of channels. So let's get that one. Okay, let's get another one. Okay. So we're talking about now the action potential. So we're going to draw them the action potential. So what is the action potential? So action potential now is a type of potential difference that's going to happen when a nerve cell is stimulated. When a nerve cell is stimulated, that's why I'm going to see what? action potential, the one that we're going to plot here. Okay, so now what happens is like this. Okay, so what happens is like this. Let's say here we don't have any stimuli, but here we having any stimulus. A stimulus can be anything. It can be maybe pain stimulus, okay? It can be maybe light stimulus, a lot of stimulus, mechanical stimulus, pressure stimulus, a lot of stimulus that can be there. So let's say that stimulus is there, then you try to stimulate this cell. Okay, if you try to stimulate that cell, something important is going to happen. Okay, the moment you stimulate this one, now the first important thing is that that resting membrane potential is going to do something. Okay, it's going to start shooting up. Then that important channels that are going to open. Okay, important channels that are going to open. This is what we call the sodium gated channel. So these are what we call sodium voted channel. So the reason why they put voted, so sodium voted channels. So sodium voted channels, because they are sensitive to voted, they change in voted. So these are what we call sodium voted channels. So the moment you try to stimulate that one, these sodium voted channels, they are going to open. Okay, if they open like that, sodium will start going into the cell here. Okay, sodium going into the cell. Okay, if sodium is going into the cell now, we're trying to we are stimulating. So if sodium is going to the cell, what are you making the inside of the cell? The inside of the cell is going to become more positive. Now let's come here so that we fully understand. If I explain something what happens initially, then we're going to plot it here. Okay, so what you are saying is remember that what is negative 70? This is negative 70 millivolts, okay, on action potential, and we say that is the resting membrane potential. So this is it. Okay, so let's just use red. Okay, this is the resting membrane potential. It is just like that. Okay, now if it is just like that, then you try to stimulate that one. If you try to stimulate this cell, then it will start moving up like that. Moving up, what does it mean? It means that you are making this cell to become more positive. From negative 70 going upward like that, you are making the cell to become more positive. It means that sodium is going in. So what happens where this one trying to reach this negative 55 that we'll talk about, the sodium channels are open, okay? They are open, they are flowing into the cell, okay? If this is happening now, what do you think next is going to happen? It's going to reach this negative 55. The moment it reaches that negative 55, what do we call this? So this negative 55, it is called a threshold potential. Very, very important. So I'm also going to explain this one. Threshold potential. Okay. So threshold potential. So that is the threshold potential. So threshold potential, what does it mean? It means that this is a type of potential whereby if a cell reaches that potential, action potential is going to arise. So action potential can only be there if you hit about negative 55 millivolts in nerve cells. So that's what it called threshold. Threshold is a minimum type of voltage that is required for action potential to be produced. Okay, so the moment now you hit this threshold potential, because the sodium channels are open, the moment you reach there, now what's going to happen? More sodium channels are going to open. More sodium channels are going to open. The moment you reach the threshold potential, more sodium channels are going to open, so that now more sodium can rush into the cell. More sodium ions can rush into the cell. Okay, so here we are saying sodium channels are open for you to reach the threshold potential. But if the moment you reach the threshold potential, what do you think? More sodium ions are going to more sodium voltage channel are going to open, making the cell to become more positive. Okay, so this is what you are going to see. Okay, this is what you are going to see. So here what we are saying is that is the action potential. Now what are we saying? More sodium channels open like that. So here there were a little sodium channel that were initially open. Now the moment it reaches negative 55, more sodium channel are going to open so that more sodium can rush into the cell. Okay. Now another important thing, you've seen this one, because these are voltage type of channels. So voltage gated channels, basically they are sensitive to voltage. So remember that the moment it reaches the negative 55, the threshold, means that some sodium channels are going to be activated. A lot of sodium channels, voltage sodium channels, are going to be activated so that the arching potential can be there. But the moment it reaches what? 30, this is positive 30 millivolts. Let's put positive. Okay. So the moment it reaches now positive 30 millivolts, okay, positive 30 millivolts, now another channel is going to be activated, because there's another important channel that is sensitive to positive 30 millivolts. What we call that? This one. This one is what we call potassium voltage gated channel. Potassium voltage gated channel. So they are going to open. Okay. Now, if these potassium voltage gated channels are opened, like what do you think? Potassium will start leaving the cell, not going into the cell, but leaving the cell. Now, something, so the moment it reaches here, the positive 30, the potassium voltage gated channels are going to be open, and potassium will start leaving the cell. If potassium is leaving the cell, it is leaving the cell negative. But you need to understand to say, what about the sodium voltage channels? If they reach positive 30 millivolts, what's going to happen to them? They are going to be closed. Okay, so these ones are going to be closed. They are going to be closed, but we are going to open the potassium voltage gated channels. So here, if that happens, since now you are making the cell to become now negative, what do we think now? Are you going to start having the depolarization phase, or the arching potential to be going upward? No, it will start going downward. Why? Now the cell, the inside of the cell is becoming more negative, okay, or negative, I mean. So here it will start going downward, okay, downward like that. Then it will also pass the negative 55 up until it reaches this one. It's going to reach negative 70 millivolts, which is also the rest membrane potential. But something else is going to happen. Okay, something else is going to happen. So the moment it reaches negative 70 millivolts, something else is going to happen. It will pass surpass the negative 70. So it will pass this resting membrane potential. Okay, so let us use blue. It will pass this resting membrane potential like that and become more negative. Maybe it can reach up to what? Let me just say negative 90. So it can reach up to what? Negative 90 millivolts. So the moment it's coming down like that, it will surpass the what? Negative 70 and go beyond negative 70. Basically, it can reach up to negative 90 millivolts or maybe negative 80 millivolts. Now the question that you can ask is that why passing the resting membrane potential? Because that's the normal voltage of what? A cell that is not stimulated. Why does it do that? The reason why it does that is because of something else. Okay, this potassium, the moment you hit the negative 70, basically we expect the potassium channels to what? To close. So it will take time for these potassium channels to close. Okay, so these voltage gated potassium channels are what? It's very, very slow when it comes to what? Close. No wonder it will pass through the what? Negative 70 up to what? Then it will reach up to negative 90. Now the moment it reaches negative 90, something else is going to happen. Now there will be this phase. Okay, there will be this phase. Okay, it will start going back to the what? Normal state like that. Okay, which is the resting membrane potential. So it will start going back like that. Okay, it will start going back to this. Okay, so now let's summarize something. Okay, so we need to summarize something here. Okay, so now since we've talked about how it happens, now you need to put in two phases. So because if you don't put in two phases, you can get confused. Quite right, you know, this stage this is what is happening. Let's say this is what is happening. So we need to put them into what certain phases. Okay, so what are we saying? We need to put everything that we're talking about into what phases. Okay. Okay, what's the first phase? The first phase is what we call what resting phase. What is the resting phase? Resting phase, we're just talking about what resting membrane potential. Resting membrane potential. This is the resting phase here. Okay, this is the resting phase. This one. Okay, this is what we call the resting phase because it is comprising of what the resting membrane potential. So that's the first phase here. Okay, the next. Okay, let's talk about now the depolarization phase. Depolarization phase. Okay, depolarization phase. So depolarization phase now. This is the first one is the threshold phase. So let's put. So we forgot one. So the other important one is what? The threshold phase. Okay. So this is the first one. The second one, what happened is what? The threshold phase. So under the resting phase, do you remember what was happening? Resting phase, that's why you can have now, you are not having the sodium channels opening. No, there is no sodium opening there, so there is no sodium opening here. But the sodium opening, gated channels opening will start from where? The threshold potential here. So the threshold phase here, that's why I think the sodium, but a little of sodium channel, okay, opening. So that's what we said there, but they are not a lot here. Okay, so that's what happens. Under depolarization phase now is what? Where sodium channels now, a lot of sodium channels, they are going to open. Okay, sodium gated channels open. So a lot of them. Okay, opens. A lot of them here. Depolarization phase. Then what's the next phase? So this is number 3. The next phase is what? So let us look at it. This is the first phase, second one, and this is the third one. You need to understand. So number 3 is what we call depolarization. So starting from the threshold up to the the peak there, that's what we call depolarization phase. Then number 4. Okay, number 4 is what? This one. Starting from here up to the resting membrane potential. What we call that? We call it what? Repolarization phase. Okay, we call it what? Repolarization phase. Okay, we call it repolarization phase, and you remember that repolarization phase is comprises of what? Potassium channel opening. Okay, potassium channel opens. That's what we say. Okay. Now, what is the other important phase? The other one is what? This one. The other important phase is what? This one. So let us remove this, make space. The other one. So here we are saying this is what? Repolarization phase. Now up to where? Up to here. But as you can see, it can reach now, this -70 can be surpassed by that, what we call now that phase, the last phase, which is what? This one. Okay, what we call the last phase, which is this one is what? Now it is too much, hyper. We can say hyper repolarization phase. So hyper repolarization phase, basically, it is now hyperpolarization is now becoming more negative compared to the resting membrane potential. That is the fifth stage. Then the last one is going back to the normal state. So the last one now is it's going to go back to normal state. How? Because here on number 6 here, that's where now that -90 millivolts will go back to the -70. Why here? It is because of the sodium potassium ATPase, they are now active, so they are working. They will make sure that action potential goes back to resting membrane type of potential. Those is sodium potassium ATPase. Okay, it's a lot. So that's all about the action potential. You need to understand about it, okay? Then the last, okay, now let us finish up. Okay, so now apart from understanding everything, now we need to understand some certain laws that are there when it comes to what the action potential, the nerve physiology, everything about it. So we need to understand about the laws. So let's start with the first common law, which we call or non-law. So this one, let's start. Let us use red. So this one states what? If a nerve is stimulated, will a full action potential or so we're saying or non-law states that if a nerve is stimulated, if a nerve is what? Stimulated, will produce a full action potential or not produce action potential at all. What does it mean? So they are saying like this. So if this is okay, if this is the action potential, this is the action potential curve, okay, like that. Let us write it well. Okay, so this is what they are saying like that. Okay. So this law states that if this is the threshold, so remember that this is the threshold. So this is the threshold. So they are saying that the moment you stimulate this nerve cell, the moment it reaches the threshold, the moment you stimulate this cell, then the moment it reaches the threshold potential, is going to produce what the action potential. But the moment doesn't reach the threshold potential, it's not going to produce what the inaction potential. Make sure that you understand. So regardless what you do, the moment you hit the threshold potential, is going to produce the full action potential. So basically, this law, if you want to understand it, is that the stimulus. So the strength of the stimulus doesn't predict the action potential. Okay, so the action potential doesn't depend on the strength of the stimulus. Okay, as long as, so let's say if we got two types of stimulus, we've got a weak stimulus and a strong stimulus stimulating the same cell. Okay, if the weak stimulus reaches the threshold potential, okay, as well as the stronger stimulus reaches the threshold potential, basically they are all going to produce the same action potential. So they are going to produce the same action potential regardless of the stimulus, as long as they hit the threshold potential. So that's all non-law. You need to understand about that, okay? Then another one. So this is the first law. The second law is what? Law of conduction by direction. So law of conduction says that if you have this neuron, okay, if you have this neuron, instead of maybe stimulating from this cell body, or maybe stimulating it from maybe the axonal terminal, you decide to stimulate it from here. Okay, you stimulate it from the axon there. Okay, if you stimulate it from the axon there, it has shown that the action potential can go in this direction and also can go toward the cell board. Toward the cell board or toward the axonal terminal. That's bidirectional. So this is the law of direction. Okay, law of that conduction, which is bidirection. If you try to stimulate the nerve fiber at any point, the direction of the nerve impulses, they will be in the bidirection. Basically, the one will go to the cell body and the other one will go to the axonal terminal. That's the law. Okay. What is the another one? Another one is this one. Okay. Another one apart from this, Bell-Magendie principle. So Bell-Magendie principle, now this one talks about the type of the type of the nerve cell that is involved. Remember that if you talk about the functional classification of nerve cells, what did we say? We say that there are what we call motor neurons, there are what we call sensory neurons, there are what we call as well interneurons. So it will depend the direction of the what? Of the nerve impulse or the action potential to depend on the type of the what cell. That is what Bell-Magendie principle. Let's get one good example. Let's talk about the sensory neuron. So the sensory neuron, what could be the direction of the action potential there? Remember that the sensory neuron takes information from the what? Receptors up to the what is central nervous system. So this one is going to take what? Information from the receptors to the what? Central nervous system. So that's the direction. So it will depend on the type of the cell. Motor neuron, remember that? Motor neuron to take information from the CNS up to the what? Effectors such as what? Muscles, glands, like that, etc. Okay? So that is what? Bell-Magendie principle. It states that the direction of the what? Of the nerve impulse or the direction of the action potential, it will depend on the type of what? Of the cell involved or the nerve cell involved, according to the functional classification. Okay? So now let's talk about another one. So apart from this, so there is what we call, I'm just going to put this one, the summation, law of summation. Okay, the law of what? Summation. Another one apart from the Bell-Magendie's principle, the law of conduction or Onani law. There's also the law of summation. It has been shown that, so we use this same one. Okay. So law of summation, basically they are saying there are some stimulus, stimulus that can't reach the what? The threshold what? Potential. So we are saying about this one. So there are some stimulus that can't reach the what? The threshold potential. Okay. So let's say if this is a cell, if this is a nerve cell like that. Okay. If this is a nerve cell and there is also another what? Nerve cell which is here. Okay. Another nerve cell which is here, but this nerve cell is failing to what? To stimulate this other what? This other nerve cell. Why? Because the type of stimulus that is there is weak. Okay, the type of neurotransmitter that is there is not enough to reach the threshold potential. So basically, what we do is that if initially it produced about what? Maybe about 5 neurotransmitters. I'm just giving an example. About 5 neurotransmitters, and these are not enough. Again, to produce another what? Another what? Neurotransmitter, which is maybe about what? 5 neurotransmitters. Together, they're going to make about what? 10 neurotransmitters, which are enough now to stimulate which one? Which are enough now to stimulate another what? Neuron. That is the law of summation. But there are two types of law of summation. The first one is what? The first one we call it what? Spatial type of summation and also temporal. Temporal type of what? Summation. If we talk about the temporal one, we're talking about this good example that I gave. The same neuron is trying to produce what? A lot of neurotransmitters so that it can sum up so that now it can reach the what? Threshold what type of potential, and so that you can have now the full action potential. Okay, that is what? Temporal. Temporal means that a single neuron basically is producing what? The neurotransmitters that are adding up to produce the full action potential. Now spatial, it basically means like this. This first neuron is failing to what? Stimulate this one. So this first neuron is failing to stimulate this other neuron, but there will be other neurons that are helping. Okay, other neurons that are helping. This one was producing this neurotransmitter. Okay, another one is producing another neurotransmitter. Okay, this one was not enough, was producing about 5. Another one is now producing about what? 5 neurotransmitters. Together now you can sum up and produce the full action potential. If the neurotransmitters are coming from other neurons like that, then they are coming together to produce the full action potential. That's what we call now the spatial type of what? Summation. Make sure that you understand. Now let's conclude with this one, the propagation of what? Action potential. So propagation of action potential, it has 2 important types of propagation. The way it goes along the what? The neurons. Okay. If you go back to the neurons classification, we had one type of classification which we call according to the what? Myelination, or if it is myelinated or not. If it is myelinated, the type of neuron is what we call myelinated neurons, and Myelinated neurons are a lot of neurons compared to what the ones that are not myelinated. But if you remember, what's the function of the myelin sheath? Myelin is important in what? Increase the velocity of what? Conduction of electric impulse or the nerve impulse. So if we talk about the continuous conduction under propagation of action potential, the continuous conduction happens in what? Happens basically in an myelinated what? Neurons. Okay, it happens in myelinated neurons, and it is a very, very slow type of what? Conduction. Why it is very slow? Because in unmyelinated neurons, those neurons, they don't have myelin. If they don't have myelin, they are slow conducting. Okay, they don't conduct at a faster rate. Okay, what else? So tactile type of conduction. Now, so tactile type of conduction, it happens in what? Just the opposite. Myelinated neurons, okay, and these are what? Very, very fast. And remember that myelinated neurons, these are a lot of neurons that we now see. They can be found everywhere in the central nervous system, peripheral nervous system, a lot of places. That's why you can see myelinated neurons. And the type of conduction that happens in myelinated neurons is what we call saltatory conduction. Okay. And there are also other important things that you need to understand. The fatigue. If you look at if a neuron is overworked, if it is overworked, basically it has been stimulated for a longer period of time. Repetitive type of stimulation can cause fatigue of neurons. Fatigue of neurons mean that a neuron can't respond to further stimulation. Okay, so then it's going to become fatigue. It will not respond to respond to any stimulus. Okay, that is another factor that can affect the conduction of nerve impulse across neurons. Okay, so that's why we end from. All the best. Make sure that you study more, and don't forget to subscribe and like the video. All the best.